actually genetics is one of the reasons why we were so interested in the glial cells. And by we, it's the field of Alzheimer's disease. So from GWAS studies and transcriptomics, as well as morphological studies, we've known from a while ago, but more convincingly from the GWAS studies in the more recent years, that a lot of genes that are risk factors for late onset Alzheimer's disease are expressed by glial cells, like microglia, for example. And one of these that is very popular nowadays is TREM2, expressed by monocytes and microglia, as well as APOE4. APOE4 is the greatest genetic risk factor for developing late onset Alzheimer's disease. And the main synthesizers of APOE in the brain are microglia and astrocytes. So we're not really sure how these might be driving some of these differences. What we do know from our study is that we were able, using some fancy statistics with the linear mixed effects model, to take into account what is the APOE genotype of the individuals that we were looking at when we were looking at the human postmortem tissue. And what we found was that both microglia and astrocytes in people with the APOE4 genotype were ingesting more synapses than people with the APOE3 genotype. But there are many differences depending on whether this is in control individuals or Alzheimer's individuals, what brain area you're looking at, whether plaques are present. So that's where things get a bit more complicated. But definitely overall, we see that APOE4 in both astrocytes and microglia is associated to more synaptic ingestion.